The overall goal of the following experiment is to deliver macromolecules vector-free to target cells using mechanical disruption. This is achieved by first placing the target cell type in suspension with the desired delivery material and loading it into the reservoir of a specially designed microfluidic device. As a second step, the microfluidic device is pressurized and the cells are driven through an array of microfluidic channels that squeeze the cells to facilitate transient membrane disruption. Next, the cells are allowed to incubate in the presence of the target delivery material to facilitate diffusive transport of the material into the cell cytoplasm while the cell membrane is repaired. Results are obtained that show effective delivery of macromolecules through the use of flow cytometry.
The main advantage of this technique over existing methods like electroporation and liposuction is that it's capable of addressing challenging cell types and materials that cannot be effectively delivered by other methods.
This method can answer key questions in the biological field, such as how to improve cell reprogramming.
The implications of this technique extend towards the therapy and diagnosis of cancer, because the system enables manipulation of immune cells to enhance their anti-tumor efficacy.
To begin, fabricate or purchase the microfluidic device, device holder, plastic fluid reservoirs, and O-rings shown here, and listed with details in the accompanying text protocol. Then, fill up 3 sealable containers part-way with 70% ethanol. Place the devices in the first container, the reservoirs and O-rings in the second container, and holders in the third. Then, fill the containers the rest of the way with 70% ethanol. Next, place the container containing the reservoirs and O-rings as well as the one containing the holders in an ultrasound bath for 5 to 10 minutes before use. This will help to remove any contaminating particles from previous experiments.